at Colby College. Oh, yes, we're being recorded. Okay, so I'm currently a postdoc at Colby College in Maine, but the work I'm going to be talking about today, I did as part of my PhD thesis at UC Santa Barbara. And so before I get into it, I just wanted to acknowledge my advisors and thesis committee and many other people at UCSB and in California that helped with this, and actually all over the country, and my funding sources and many volunteers who, without this, I couldn't have done this work. So this whole series is on kelp forests. And so if you've been attending the other talks, you've learned some things about kelp forests. And Kyle's work often looks at kelp forests from the surface, where you see this dense floating canopy. And if you have maybe been to the Monterey Bay Aquarium or seen any illustrations of kelp forests, you may have seen this romanticized illustration of what a kelp forest looks like and all of the organisms in it. And if you've been diving or maybe seen some underwater photography, maybe you've seen these beautiful underwater views of kelp forests. But if you've done a lot of diving in kelp forests, you probably are more used to seeing them look something like this, where they're very murky, you can't see really what's going on. And I think that this is kind of a good representation of the complexity of kelp forests and how we can't actually see most of the interactions among the species in kelp forests. So in an attempt to describe this complexity, people have made food webs for kelp forests. And a food web is a map of all of the feeding interactions among species. So this is an example of a very simple food web that is a cartoon version, and it shows some of the larger species and how they're connected. Scientists have tried to improve upon these and have made additional food webs that have higher resolution in some cases, but often they've still lacked high resolution for some of these larger groups, or they've been missing some really small organisms or really camouflaged organisms, or those organisms that move throughout the system and maybe don't hang around too long. So my goal for my PhD was to build a better food web for the kelp forest to sort of describe all of this complexity that was happening in the system. And again, a food web is simply a map of who eats whom in the system. And there are lots of ways you could try to learn about who's eating whom in the system, but oftentimes you don't see these actual feeding interactions. So we might know that sharks are eating fish, but often when you bring up a fish from the ocean and dissect it and look in its stomach, the stomach is often empty. So it can be hard to know what they're actually eating. But parasites can actually help us get at this, because parasites can be transmitted through food chains. I'm just going to give you a quick definition of what a parasite is, and this is the way I'll be using this word through the talk. So a parasite is a consumer that lives on or in another organism, so we refer to that as the host, and it eats its host and it causes harm to that host. So there are some organisms that live, species harm those hosts. We can call those mutualists or commensals, but we're going to be focusing on parasites today. Many of these parasites have complex life cycles. So this is an example of a complex life cycle that most people are at least sort of familiar with. So a butterfly lays an egg on a plant. That egg hatches into a caterpillar, which eats the leaves, and then it goes through metamorphosis, becomes an adult butterfly, and this is a complex life cycle. Many parasites also have complex life cycles that often require different host species at each stage. Often they're transmitted between through feeding interactions. So we can use these to help learn about feeding interactions because they accumulate in hosts over time. So we can actually infer things about predators and prey. So for example, in the photo shown here, there's a larger fish with an adult tapeworm. It passes eggs out of its gut through its feces. Those eggs are eaten by a small crustacean and develop into a larval stage. A smaller fish eats that crustacean and gets this next stage of the parasite, and then that is finally eaten, final host, and the parasite develops into its adult form. So by finding these parasites, we can learn about what the fish was eating. So this is just another example of this. I just described the same thing in the previous slide. So we find a shark with a tapeworm in it, and then we can learn about what the shark was eating. Or alternatively, when we find smaller fish and prey items with shark tapeworms in them, that tells us that those sharks are in the system and actively there because these fish are getting infected with their parasites. Other people have realized parasites are important in food webs, and where researchers have looked for parasites in food webs, they found that they actually make up quite a large proportion of the species in the food web. So this graph is showing marine food webs. The blue bars are showing the number of free living nodes, and I'll describe what a node is specifically in a moment, but basically, usually it refers to a species in the food web. So this is the number of free living nodes. And then you can see that where we've included parasites is these red bars. They make up a large proportion of the total number of nodes in the food web. So I focused on the Santa Barbara Channel, shown here on this map. And I collected data from basically any source that I could. So we did reviews of the primary literature. We used data sets from long-term research groups. We used crowdsourced observations. We used technical reports. We used guidebooks, and we talked to experts on the topics to make sure that we weren't missing anything. And sometimes people have unpublished data on things that they're willing to share with you. And then where we had missing information, we collected data ourselves. So we did zooplankton tows at several sites, and we identified things to species where we could. We also sampled kelp holdfast. So that's the way that the giant kelp attaches to the substrate, and we sorted the organisms there. And we found some pretty interesting things. So this top, I don't know if you guys can see my mouse, but in the top right image, that's an adult male of a, actually, I'm going to go back one slide. So in the top right here, this is a type of isopod that is a blood feeding organism. It's kind of similar to a mosquito, where it swims around and sucks blood of fish. And guy on the top left on this slide is the adult male of that same type of organism, but it lives in kelp holdfast, and it is not blood feeding. It just is, they think it eats detritus. And so that was pretty interesting. And then we also collected samples of understory algae and looked for all those small gastropods, mollusks, nudibranchs, snails, and things like that. And then we collected a lot of organisms and looked for parasites. So we collected 48 species of fish, including 5 elasmobranchs. And you could see how much my hair has grown since I did this work. And then we collected 85 species of vertebrates, over 2,500 total dissections. And we would basically take these animals apart and squash the tissues between glass plates and look for all life stages of parasites. Many published parasite studies focus on adult stages, but we were really interested in those larval stages as well, because they helped us determine those feeding interactions. So then we actually started assembling the web. So here I have my cartoon of a shark and the fish. So the first part of food web are the nodes, and these are just the organisms in the web. So if we were going to be focusing on species, our shark species would be one node and the fish species would be another node. And then we assigned links, which are the connections between the organisms. And in the case of the food web, those links represent a feeding interaction. And that is usually just represented as just an arrow pointing between the two. When we built this, at first we included life stages as separate nodes if we knew those life stages were all present in the food web and different enough to be considered different. So for example, an adult rockfish and a recruit rockfish have different feeding interactions. They have different predators. And so we included those as separate nodes. And we did the same thing for parasites. So an adult parasite and the larval stage of that same parasite were included as separate nodes. Then we started assigning all the links between them. So we used direct evidence of links. Those were kind of the easier links to assign because we saw it ourselves or we had records of it in the literature. And when we didn't have direct evidence, we used indirect evidence and logic to do this. And so this is kind of the steps that we went through. So we used two categories. One was whether two species were likely to encounter each other. And the other was, if they did encounter each other, how compatible would that feeding interaction be? So the encounter one is important because if two species never interact in the habitat, they're not likely to feed on each other if they never come across each other. So for example, if you have a fish that typically hangs out in the kelp canopy, it's unlikely to be encountering a snail that lives buried in the sediment, for example. And then compatibility are things like, do they have the same mouth parts that would actually allow them to consume it? Are they the correct body sizes to feed on each other, etc. So examples of things we thought about with encounter: what microhabitats are these animals living in? What times of day are they active? What is the feeding method? And then for compatibility, we thought about body sizes, possible defenses of the prey or the host, how host specific are these parasites? Maybe we know from studies of them that they only infect one host species, etc. And if we had evidence that they both encountered each other and they were compatible, we assigned a link. So as an example of how we actually would use this parasite information to learn about predator-prey interactions, this is the Senorita wrasse. It's one of the most common species of fish you'll see in Southern California kelp forests. And we found this parasite here in its fins fairly often. It's an encysted stage. And when you get a tiny little pin and remove the worm from the cyst, you get this little worm. And this is a type of fluke. The technical name is a trematode. And you can zoom in on it and you see it's got all these spines on its outer surface and some hooks. And this parasite as an adult is really only found in moray eels. And so we were able to use that information to say, okay, moray eels are likely around and feeding on Senorita wrasse. So when we do this and we go through and we add parasites and we add all these feeding interactions, we found that when you add parasites, it really leads to 4 different types of interactions in the food web. So the first type is a predator eating a prey. And then we can add in our host parasite interaction. So you probably have already picked up on this, but I'll be showing parasites in red and free living species in blue. And then there's this third type of interaction where a predator can eat a free living species and in doing so also consumes the parasites of that prey species. And so if the predator is not a suitable host for that parasite dies, and that's called concomitant predation, and it's an important source of parasite death. There's a fourth type of interaction where parasites within a host can attack and consume each other, but I didn't observe this in this study, and so I don't have any of those types of links in this food web. Okay, so now I'm going to be showing you the full food web, and I'm going to be building it from the bottom up, and things are going to increase in trophic level. So the first layer down here at the bottom are the producers. So this is algae, phytoplankton, as well as detritus links. And yeah, I won't get into the detritus links, but there's a few different categories, but you can't see everything because the way it randomly laid it out, some things are overlapping. And then here are the free living species shown in blue. And so you can see this first row of blue dots all in a line. Those are the primary consumers. So these are pure herbivores that are only eating algae or detritus, essentially. And then everything else is some level of omnivory. And then we can add in all of the links between the free living species and their prey sources. And then here are all the parasite species shown in red. So you can see that they're just kind of this cloud of parasites hanging over all of the free living species. And then here are all the links between the parasites and the free living species, so their hosts. And I didn't add in that third link type, the concomitant predation here, because this graph is already very crowded and adding in the concomitant links, you can't really see anything. And so if we compare the food web with just free living species and with parasite species added, it almost doubles the size of the network. Just letting the images load, because these pictures are large, so Zoom sometimes gets upset about it. And it more than doubled the number of links. And if we break down the link proportions by link type, we can see that the most common link type was actually those predator-parasite interactions, whereas there were fewer parasite-host interactions than predator-prey. And we'll talk a little bit more about why that is in just a few slides. When we compare the number of parasite species in kelp forests to other food webs where parasites have been included, we can see that parasites added more species to kelp forest food webs than to these other ecosystems. So this figure shows these other habitat types. So along the x-axis, you'll see there's an Arctic lake food web, an inner mud flat food web, some estuary food webs, and then a tropical sand flat food web. And then the y-axis is the number of species, the blue bars are the free living species, and the red bars are the parasite species. And we can look at this by proportion as well. So this is the same data, just shown as a proportion of the total instead of as absolute numbers of species. And here it's a little easier to see that parasites actually made up a larger proportion of the total species in kelp forests compared to the other food webs. Can break down that species richness by type of organism. So this figure on the y-axis shows the group of organism, and this is not a formal taxonomic distinction, but it refers to kind of the way people tend to think about these different organisms. And then along the X here is the species count. So the trematodes, which are the flukes that I mentioned, had more species than any other group. And the second most speciose groups were the crustaceans, and there were actually more parasitic crustaceans than free-living crustaceans. So again, the trematodes had the most species, and I found a lot of adult trematodes. These are just some examples of some of the species we found. And I know that the first time I saw pictures of parasites, I didn't know what I was looking at. So I just wanted to show you guys some photos and explain what they look like, because I think they're very beautiful and have very interesting anatomy. So on the top, on the left, let me see if I can get like a cursor here. Okay, here's the laser pointer. So in this worm here, at the top is the end with the mouth, and then it has a sucker on its ventral surface. And then this swirl here is actually the uterus. And this is one of my favorite trematodes because it's really easy to identify. And all of my volunteers also loved it because they're like, oh, it's that one. So it has this kind of tornado of eggs as its uterus, or in its uterus. And then it has 7 to 11 testes here. And then all of this dark coloration on the sides is yolk producing organ. So because parasites don't really have to worry about where they're getting their food or protecting themselves from the elements, they often are just entirely filled with reproductive organs, and they can just dedicate all of their energy basically to reproducing. And so these other pictures are just other species, and they have all the same organs, but they're just organized in different ways. So the parasitic crustaceans had the second most number of species, and there were more parasitic crustaceans than free living species. And most of these were parasitic copepods. And I just wanted to show you some of the differences between a free living copepod and a parasitic. So on the left here, these photos are of, and these are not scaled, so don't try to compare sizes, but on the left here are free living copepods, and on the right are just 2 examples of parasitic copepods. And you can see that the parasitic ones have these hook-like raptorial appendages that they use to latch onto the host. So most of these were actually all of them were ectoparasites, often on the gills or the fins. whereas the free-living copepods are planktonic, or some of them are living on substrates, but they tend to be much smaller and they don't have any kinds of crazy hook-like appendages. Parasitic copepods also tend to be much larger than free-living, which is a phenomenon that happens in multiple groups of parasitic organisms that have free-living counterparts. Sometimes the parasitic versions of things are larger. So how are all of these species connected in the food web? And one of the ways to think about this is to look at the numbers of links connected to each node. And so in this food web, that includes both the prey and the hosts, or the prey or the hosts of a species and that species' enemies. So for example, this blue node here would have 2 prey species, and it would have 3 different enemies, 2 of which were parasites. And so if we break this down for the whole food web, shown here on the left, this graph is showing the diet breadth of free-living species. So this is the number of prey that each free-living species has, and the count of prey species is on, or the count of species is on the Y. And so you can see that there's this long tail. So all of these species have prey items, whereas these peaks here are species that are either specialists or producers. So this bar over the 0, those are producers that don't have any prey items. And then on the right here are the parasites. And so we have a sort of similar shape, but there's a more slender tail, and there's this strong peak at 1. So these are all parasites that only have 1 host, and then you have another chunk that have 2. But you also see some parasites that have many different host species. Most of these parasites that had many host species were generalists that we found a lot of larval stages for, and they were basically insisted in hosts waiting to be eaten by the correct final host. And so I wanted to walk you through some of those, because these are basically the parasites that are interacting really strongly in the food web. So one very common group that we found were thorny-headed worms, and the pictures shown here are the encysted larval stages. And these typically go into warm-blooded vertebrates, or I guess all vertebrates are, or all warm-blooded animals are, well, that's not true. So typically going into birds or mammals. And then we also found a lot of nematodes that went into mammals and birds as their final hosts. And we found a lot of shark tapeworms. So these groups of tapeworms here only use sharks and rays as their final hosts. And so I wanted to walk you through how these parasites are moving through the food web. So in this example, I have a pile perch, and we found a lot of larval stages of shark tapeworms in this pile perch. So this picture here is an encysted larval stage, shown a little closer. If you can extract it out of that cyst, you see the attachment organ here. And they have these very impressive hooks that they can extend. And once they're in the shark, they use these hooks to anchor into the intestine. And this particular parasite uses leopard sharks, most likely, as its final host. So we could put these links into the food web. So we have the larval stage in the pile perch. If the pile perch gets eaten by the shark, the tapeworm develops into the adult. But there are a lot of other predators in the kelp forest. So this fish could be eaten by a giant sea bass or a lingcod or any other number of piscivorous fish. And in those hosts, that parasite has the ability to re-insist and remain in that larval stage. In some cases, that might be a dead end. So if it ends up in a black sea bass, a leopard shark is probably not going to take down a black sea bass. But if it ends up in something a little smaller, say a rockfish or a younger lingcod, maybe that leopard shark could eat it and get infected with the parasite. So this is called a paratenic host, and this seemed to be very common in kelp forests. So another example, I'll just go through this really quickly, but it was one of the most common parasites we found, so I wanted to show it. This is a larval stage of a thorny-headed worm. This is what the cyst looks like. When you extract it from the cyst, this attachment organ here everts out and reveals this hooked proboscis. And many parasites don't do too much harm to their host if the intensity of parasites is low, but these guys can actually cause some harm to the host because you can see how intense this hooked appendage is. This particular species uses harbor seals as its final host. And we found this larval stage in almost every species of fish we dissected. So it was really throughout the food web. It uses amphipods, which is a type of crustacean. I think my thing froze. I don't know. Oh, there we go. It uses amphipods as its first host and many different species of fish as paratenic hosts. So I wanted to show you how some of these now are embedded in the full network. So here's the food web again. I've just colored everything gray, removed the links. So this Corynosoma stromosum is I'm just gonna get rid of the pointer. Whoops, sorry. So here is the larval stage of this parasite, Corynosoma stromosum, thorny-headed worm, and it had 58 fish hosts that it could use in the food web. The adult, although it does the best when it's in a harbor seal, it can survive and reproduce in basically any warm-blooded vertebrate. So it can survive if it ends up in a seabird or another marine mammal. And there were 16 of those in the web. If we draw lines between those final hosts and the intermediate hosts, that reveals that these are feeding interactions between those final hosts and intermediate hosts. There are 339 potential transmission pathways for this parasite to complete that step in its life cycle. We can do the same thing for some of the other really common generalist parasites. So this is Contrasecum. It's a nematode or roundworm of seabirds. And it also had 58 intermediate hosts and the same 16 definitive hosts. Or not the same, but some of the same definitive hosts, and it had 239 opportunities for transmission in the food web. This is a shark tapeworm that was found in many different fish species, 68 intermediate hosts, 10 possible final hosts or definitive hosts, and 147 transmission pathways. And so you can see that these parasites are really embedded in the food web and are taking advantage of all of these interactions between the intermediate trophic levels and the top predators to complete their life cycle. So again, these really densely connected parasites are using these trophic interactions to survive and reproduce. So with this food web completed, we can add the kelp forest food web into this plot that I showed you at the beginning. And we can see it here at the top. It's the largest marine food web on this graph, at least, and one of the largest ones that I know of. And it has this large proportion of parasites in it. And it just adds to this, adds support to the idea that parasites are an important part of food webs in general. So if you take anything home with you, my main things I want people to think about are that parasites are utilizing food chains to complete their life cycles. And so they're really interwoven within the food webs. You know, in the beginning, I showed you that they kind of just looked like this cloud on the surface, but they're participating in all of these interactions between the free living species. And using parasites helped us determine the feeding links to complete the food web in general, not just adding parasites to it. They helped us learn about the free living species interactions too. So now if you see a kind of romanticized food web drawing again, or a kelp forest drawing, I'm hoping that you'll start imagining that all of these animals are just filled with cysts of flukes and trematodes and tapeworms and nematodes and acanthocephalin thorny-headed worms and parasitic isopods and parasitic copepods. And there's other things that I didn't even talk about in crustaceans. And so with that, if we have time for questions, I'm not actually sure how long I talked for, but I'd be happy to take any questions. And my new email here is at the bottom. So thanks.